We have got a lot of questions coming in. Some of them are very general, but I guess we can get started with some of the general ones and then on to the more specific ones. So in the beginning of your slides, you had talked about a lot of microbial resistance, but I think one of the audience members have a question. What is the difference between drug resistance and drug persistence? Could you elaborate upon that?
Right. So actually a very good question, in fact. So the issue with drug resistance is that, so I'm not sure if you have read this or seen this video. So if there is a drug resistant microbe, I've shown you, right? If there is a population and you give a drug, the entire susceptible population dies, and the ones that remain are the ones which are resistant to the antibiotic. In a persistent scenario, if I continue that same thing, and if I now expose the golden buns to toxin molecule or the drugs again, a lot of them will again not survive. Only a subpopulation of bacteria will keep on surviving. So the issue is, these will, there is no change in MIC of a persistent pathogen, but what happens is they will actually grow, they will not expand in the presence of the antibiotic, right? But the issue is, the ones which are resistant are more genetically resistant. Like there are mechanisms that they are making it resistance to this antibiotic, and when the antibiotic is there, none of them will survive. That will not happen in a persistent population, and it's only a subpopulation. We are yet to at a molecular level understand what this is, but people say it's a more phenotypic phenomenon than the genotypic phenomenon, and the entire scenario complicates when you bring tolerance in between. I'm not doing that, but yeah, but there are differences, and I'm glad somebody paid attention to that.
Thank you for the clarification, Dr. Bhardwaj. Another question, the metabolic rerouting approach that you described to tackle TB was very interesting. Are such approaches used in identifying drug targets for other diseases, like cancer, for example?
Right. So again, a good question. I cannot cite a direct paper on that, but these are very generic metabolic approaches. The way it differs in a eukaryotic organism versus a prokaryotic one, that in prokaryotic you have a single compartment. So all the fluxes are actually between that compartment and the media in which the organism is growing. When you talk of a eukaryotic cell, which is obviously is in the case of cancer, then you know there is mitochondria, there is nucleus, and there is Golgi and ER. So the metabolic flux studies becomes a little bit more complex there, because then the rerouting of flux has to be at a different compartmental level. So if people have done that for cancer, I will really have to check that data. I have not done that, but I know the complexity that happens when you bring it to a eukaryotic system. And that's one reason I never did it. I did it on bacterial population, because, when you're learning something, you have to start with a simpler system. But to believe me, when I've been working on bacterial systems for quite some time, and they're anything but simple. So yeah, but I'm not aware of any direct studies.
Thank you. Another question, is hybrid pharmaphore similar to fragment-based drug discovery?
Yeah, so in a fragment-based drug discovery, you already have fragments. And what you're doing is, when the fragment really goes and binds to your, the target of interest, then you grow the fragment and can, make that into a full molecule. In this case, we are not really fragmenting anything. We are saying that, okay, these are the interaction features of this hypothetical molecule that we designed. And then we were trying to see which other molecules, which are the complete molecules, have similar interaction features. So we were exploring for the pharmacophore features rather than a small fragment and then grow it. So yeah, there is an in-principle difference in the way the methods work.
Okay, no science talk is complete without an AI-related question. So we have a question: with the complexity of cell structure and antibiotic treatments, have you considered using AI in your research? And we had another related question. If you are aware, could you share some AI tools that are poised to assist in such studies in drug discovery?
Yep, you're a very good question. And believe me, a lot of people in the world are working on this. So there is, I'm pretty sure if you say AI, you want to segregate it from machine learning. There are a lot of machine learning models that are already out there for a very large number of bug-drug combinations. The challenge using any AI tool is that you need very high quality data. right? If you don't have very good quality data, the AI model is going to produce some garbage. So you need very high quality data from from all geographies in the world, because what we have observed in our studies is that these pathogens are evolving in a very geography specific manner, depending on what is the kind of treatment, because most of it is empirical treatment. So what are the kind of antibiotics they're exposed to? Because in most cases, the antibiotic is not single. So the problem for AI or ML methods today is how do you generate ML or AI based methods for combination antibiotics and delineate which antibiotic triggered what mechanism of resistance. That is an open challenge right now, because everybody is developing methods for one antibiotic, which is never the clinical scenario. They have combination antibiotics, and that is where people are working towards that. For single, there are a lot of ML models that are out there. So if you type literature, you will actually find a very large number. So do that. In terms of the methods, there are actually several. People today are applying LLMs in, to understand AMR, but I haven't seen anything which is worth mentioning. But there is not to understand AMR. But recently, there has been a paper on Acinetobacter baumannii where they have identified inhibitors against AB using AI-enabled methods. So that has been done. So AB, there is a paper very recently. I can share that with you.
Thank you, Dr. Bhardwaj. I think there was already a question that was asking about major roadblocks in using machine learning in biological applications, and if the variety of data is an obstacle. I think you partially answered that. But a related question, are there any specific requirements regarding this machine learning approach to be taken when working with biological data sets? And are there some considerations or standards to be mindful of during work similar to yours?
Which question is this? Can you point me to that?
It's somewhere in the middle.
Okay.
6:30 to
Can you please
6:35.
some important considerations or standards to be mindful? Okay, so first thing is there are global standards to report MIC values. If I'm talking of AMR, the MIC values actually are the breakpoints. So you know at what MIC concentration are you supposed to designate your isolate as resistant to antibiotic or susceptible to antibiotic. If I am using an antibiotic amount of 2 micrograms and my pathogen is, or my strain is not literally getting killed at that concentration, and you are using 4, but the breakpoint is neither 2 or 4, then we are actually following a non-standard way of defining RNS. And this is very, very common in literature. That is why don't blindly use these data sets. See whether CLSI or UCAST MIC breakpoints are being applied. There are ways to look at the quality of the genome sequence data sets. UCAST has some recommendations on that. Look at high quality data to establish genotype-phenotype correlations. Do at least these 2 standards. Because otherwise, it's a lot of confusing data that you're giving to any statistical tool, and it is not going to perform well. With any amount of data, if it is wrong, you're not going to get answers.
Thank you. I guess your talk did inspire a few undergrad students in the talk attending today. So there's a question, what would be your recommendation for an undergrad student who is planning to work in this field?
Good question. So the best way to learn is to do it. So learning by doing is what I always recommend. So what you can do is, because there are pair guidelines now, which tells you that every paper has to have, findable, accessible, interoperable, reusable data sets. Pick up a paper where there is a GitHub, there is a link to the GitHub repository, the data is available, the method is available, and reproduce it. Because if you have reproduced some work, you would know how exactly it has happened. And that is the first step to learning. So do it to learn it, because there is, it's like you are asking me, how do I learn swimming? So the only way is to do a bit of theory, but then you have to jump in water and then try it out. Any amount of book reading is not going to help you swim. So you need to get in water.
Thank you. Another question, how are you dealing with off-target toxicity on host protein targets?
So good question. I skipped that in my presentation. So we actually do 3 levels of elimination of the host proteome. One is sequence-based. So we try and see if there is no sequence similarity. The second is, there are binding pockets. So there is a method that Dr. Nagasuma and IISc developed. We use that to make sure that there are no common pockets between the bacterial and the host, the potential candidate targets. And the third thing is, it has been shown that even 8 to 16 amino acid sequence is enough to form a fold in a protein, which means that something can really bind here. So what we did an octamer based exact match of an octamer library between the host and the bacterial targets to make sure that there is no overlap. So 3 methods to eliminate anything which is host-like.
There is another question that might not be as related to your work, but would be happy to receive any comments from you. With regards to drug discovery and drug production from medicinal plants, are there any researchers that is approaching the drug's efficacy with present deficiencies in the human subject? For example, the study of efficacy of nephroprotective drugs with the presence of G6PD deficiency.
Right. So I think the question is pointing to the fact that how much of natural products are we using in drug discovery? So just to give you an answer, most of the drugs that we have today that we are using are of natural product origin, most of them. There are a large number of synthetic molecules also out there, but we have been using natural products for drug discovery. The way it is progressing right now is that since microbes are now have always been considered this, the sources of our new antibiotics, a lot of genomics can be used to now find gene clusters, the biosynthetic gene clusters in these microbes to see what kind of small molecules they can make and if they can work as antibiotics. So apart from medicinal plants, that is also being done. In medicinal plants, the thing that one has to remember is that in most cases, you are going to use the entire extract. You don't know what the bioactive molecule is, and that can be very confusing. So one has to do you need to know what is the active ingredient, isolate it, and show activity with that, even in combination, but you exactly need to control what are you testing for. Because in that particular extract, you don't know what the composition is and what is really driving the activity. And then, those become very difficult to progress in a drug discovery pipeline with the current understanding.
I guess we are really coming to the end of the time. I'll take one last question. I think a very good question to conclude this science talk. Can you share what are some of the major challenges associated with big data in genomics and drug discovery?
I think a very important question indeed. So one of the first challenges that, the people working on the project. See, it's very, very important to have human resource who can think interdisciplinarity. Like say, for example, I'm a biologist, right? But I do computer programming all the time. I have to do maths every day. I do statistics every day. So basically, I'm a biologist who can do coding like a computer scientist. I know information technology. I know mathematics. Basically the reason why we have to do like that is because the nature works like that. Nature does not understand physics, chemistry, biology, or mathematics. Nature works with everything together. So if you have to understand something which is of natural origin, you first of all have to equip yourself to at least, be in a position to at least ask the right questions. So that's the first challenge. How do we train ourselves to ask the right questions? And the second challenge is, now if you have these skills, then you need to look at data set as big as possible. And the current challenges include the fact that a lot of this data is not quality controlled. We need enough experimental data which is highly quality controlled, and that is one of the biggest challenge doing drug discovery today and big data. Genomics, we have been able to do extremely well, and the reason behind that is genomics have always been open source, open access. Drug discovery, not so much. So for genomics, you know the chemistry of every sequencing platform, how the data is generated, how it has to be read, what it means. But the minute it comes to drug discovery, a lot of the steps in the entire process are blinded to the community to solve. So that kind of non-exposure to the problem itself, making the process very, very hard. And that is why genomics we did very well. In past 20 years, we literally, it's changed the way we are looking at a lot of things. Drug discovery also, if we have to do a similar, progress, we need to make sure that a lot of this data is out there for all of us to look at. And that is, I think, it's going to solve most of the problems that we are dealing with.
Great. Yes, thank you, Dr. Bharadwaj. There is a lot of love in the comments, thanking you for the wonderful talk and how everybody enjoyed this session of ACS Science Talks. As we are closing out, Dr. Bharadwaj, I would welcome you to share any final remarks or message you would have for this community present with us today.
Okay, so yeah, I'm very, very overwhelmed, first of all, with the response. So thanks you for staying late on a Friday night to listen. But I would only like to say one thing, that as members of a community, a society, we should live in synchronization and in equilibrium with our environment. Try to make sure that whatever we do is sustainable, because you can have a lot of documentation on sustainable development goals. They will remain in files if we don't act on it collectively. So understand what is your responsibility towards society. Try to see in whatever manner you can contribute, try to do that. And there is no limit to what you can, and how you can really help, how we can really help each other going forward. Because I think what COVID-19 taught us in a very, very harsh way was what happens when we are really not prepared for a pandemic of that scale. And you saw even the best healthcares in the world literally shattered. It was no country in the world that said, okay, nothing happened in this part of the world. So, and that happened because we were not working as a team. People were priming up the problem from a very long time, and nobody really paid attention. Let's not do that. Be more sensitive to what everybody else is feeling. Be more sensitive to what is required from us, and just work in that direction. And if you do it in a scientific way, there's nothing better than that. So yeah, just follow an evidence-based path to whatever you do.
Thank you so much, Dr. Bharadwaj. That was a very inspiring session from you. And also, thank you for so many efforts from your side in bridging that, apparent gap between science and society. So thank you to all of our attendees joining us today. I'm sure all of you have been inspired as much as all of us at ACS. We are really happy that we could join this community together on this Friday morning, evening, afternoon. Many parts across the globe, people have joined in today. So thank you so much on behalf of the ACS team, and wish you a good day, good evening ahead. Thank you, and take care.